worksheet number 9. So in worksheet number 9, we're going to continue kind of what we did with worksheet number 8. So if you remember with worksheet number 8, we really were working on our first worksheet that dealt with metabolism. Okay, and metabolism, as you are well aware of at this point in time, there are lots of metabolic pathways, and there's lots of details to the metabolic pathways, and those details are important. Okay, but there are also some overarching things that we need to consider as we look at metabolic pathways. And one of those things that we talked about last week is that we need to have an integrated view of metabolism. And by that, we mean that there are pathways that are connected to other pathways in the cell, and they're connected through the materials that maybe one pathway provides for another, as well as the energy that one pathway provides for another. For instance, we could think about the citric acid cycle needing acetyl-CoA. Well, acetyl-CoA is going to come from pyruvate, and pyruvate comes from glycolysis. So there's a material connection between glycolysis and the citric acid cycle. At the same time, we could think about, like, for instance, we looked at how glutamine can be made from glutamate. And to do that, you needed ATP, or we need ATP. And ATP is connected to or made from pathways such as cellular respiration. And so there's then this energetic path, but the energetic connection between cellular respiration and then the production of glutamine from glutamate. Okay, so that's one of the overarching themes of not glycolysis, but of metabolic pathways, is how they're connected materially and energetically. And so what we want to do in this worksheet is continue on this big themes and this idea of the integration of metabolic pathways. But in this case, what we want to do is consider how pathways' rates or their flux will change in response to other things going on within the environment, or the change in flux or the rate of some other pathway. And to do that, we need to think, first of all, about how pathways are designed, okay, and what that design allows for them, or how that design allows for them to be regulated. So that's the first thing that we're going to do. And then the second thing we're going to do is we're going to apply that design to a particular metabolic pathway, in the citric acid cycle in particular. And we're going to look at how then changes in particular environmental conditions within the cell will affect that pathway and either upregulate it or downregulate it so as to maintain a steady state within the cell. Okay, so let's look at activity number 1. And in activity number 1, we're looking at this hypothetical pathway that you see on your screen right now. And really what we want to do here is think about how is this pathway designed and how is that reflected in actual pathways within biological systems. And what you'll recognize here is that there are actually 2 different arrow types, okay? And those 2 different arrow types represent the types of reactions that you would find in a metabolic pathway, okay? You've got these single-headed arrows that represent these irreversible steps, okay? And those then are one type of reaction that you can find in a metabolic pathway. And then you've got the double-headed arrows, or the equilibrium arrows. Those represent reversible steps, okay? And so what I want us to do is to make an analogy of this pathway with a highway or an interstate. All of us have some experience driving on some sort of an interstate. In Colorado, right, it's I-25. And I-25, in particular, between Fort Collins and Denver, it undergoes a lot of construction, okay? And there are times where there's bottlenecks in the interstate, okay, as you head in one direction, okay? Now, understand that what we're saying here is we're headed in one direction, okay? That's the analogy. We're just going to be going in one direction. Okay, just let's say south, okay? All right, but we know that there are bottlenecks, and the reason there are bottlenecks is because possibly there's an accident, or there's construction, or there's something else, and there's fewer lanes, and then that's going to cause frustration for us, okay? Because it's going to limit how quickly we can get to Denver, okay, from Fort Collins, right? So same thing when we think about metabolic pathways, there are bottlenecks, okay? And so we want to know which one of these types of reactions are going to be the bottlenecks, okay? And how would we refer to that if we were to make a pathway or a highway of this pathway? So for instance, would our pathway look something like this? It'd start off real narrow, okay? Sorry, let me go ahead and get rid of that and try and draw a little bit straighter line. Would it start off real narrow and then kind of widen out and then go back back to narrow. Okay, dot, dot, dot. Or would it start out wide and then get narrow and then go back to wide? And so on and so forth, based off of, are we looking at a reversible or an irreversible step? Now, remember, we are, again, going in this direction. Okay, we're not trying to model north and south or whatever. Okay, in both directions, we're just going in one direction. Okay, so go ahead and take some time, think about that. Okay, pause this. And do the activity, and then come back and we'll discuss this together. I'll give you some feedback, okay? Okay, so hopefully you have had some time to think about this and you've drawn out some highway that would represent what's happening here in this hypothetical pathway, and you really had two choices. So let me go ahead and get rid of these and represent this hypothetical pathway as an interstate or as a highway, okay? And what we want to recognize here is this, that these steps here and here and here, those are irreversible steps. They are generally catalyzed by enzymes that are inefficient relative to the other steps in the pathway, these reversible ones, okay? Then I'll put these stars over here, okay, the red stars, and those reversible ones are actually very rapid. They are catalyzed by enzymes that are either in a large concentration in the cell or they're very efficient or both, okay? And that would be relative to the irreversible steps, okay? So our irreversible steps truly do then represent our bottlenecks, if you will, of our pathway. And so what we should do then here for this pathway is that we'd want to draw it something like this. Start off with a bottleneck and then expand and then go back to another bottleneck and then really expand it. Okay, and so our design of our pathway would look something like this. Okay, now the reason I like this analogy is because we all have some experience. Now, just I guess maybe before I get into that, okay, understand that this bottleneck represents the A to B step, and then this bottleneck represents both C to D and D to E, okay? And then we have here, right, these expanded sections. Actually, we can just do it this way. This expanded section here will be all that, okay? All right, so that's what we're looking at here, okay? Now, the reason I like this analogy is because we all have had experiences driving, and we all know that, man, if we could just increase the number of lanes on this highway from 2 to 4, then we would be flying right now. We would have no problems getting through this traffic jam that we're currently sitting in. And that's the same way when we look at this, okay? These bottlenecks provide these traffic jams, okay? There's only so much material that can get through what we would call our rate limiting steps that are represented in these bottleneck areas, okay? So if we want to answer a question, it says, what needs to be done to increase the rate through this pathway? Well, the thing that needs to be done to increase this rate is that we would want to expand upon these bottleneck sections, okay? Now understand that can happen by enzymatic regulation, right? Those enzymes are inefficient, but those are also enzymes that are regulated, okay? And they can be regulated by a variety of different things in the cell, okay? In particular, they can be regulated by like allosteric means, okay? So there can be an allosteric modulator, there'd be a positive modulator that then would make it more efficient, and then that would expand that particular portion of our highway, and that would become less rate limiting, right? And the flux through the pathway would eventually increase, okay? Right now, given that, what we want to do is we want to take that, the lessons we learned in that, and we want to apply that then to this pathway that you're looking at here. And hopefully you recognize this is the citric acid cycle. You've gone through the citric acid cycle at this point in time in the Echo 360 recordings, hopefully. Okay. And what we want to do here is we want to just concentrate on just the citric acid cycle. Okay. So really we want to start here at oxaloacetate and its conversion to citrate, and then we want to go all the way around. Okay. Kind of ignore this step right here. Okay, the figure has the conversion of pyruvate to acetyl-CoA, but go ahead and ignore that as we look at this. Okay, and in this activity, it's going to ask you to, first of all, linearize this pathway, okay, and draw a highway of it, okay. So based off of what are our bottlenecks, where are our bottlenecks, okay. So that's the first thing you need to do. The second thing you need to do is it says there's this figure. It says below is a figure illustrating the regulatory mechanisms of the citric acid cycle. Using this graph, redraw your highway, assuming NADH levels are dropping and ADP levels are rising. So what would happen to our pathway? Would it look different if NADH levels went up and ADP levels were going down? How would it look different, right? What regulatory mechanisms will be required, and how would that change the quote-unquote number of lanes that are available to get things through, okay? And then finally, we do one more, and it says, now that you have done this, let's see if we can do some problem solving. We will move to cell culture, which are cells in a petri dish. They're alive and running the citric acid cycle. As a researcher, you infuse an inhibitor electron transport chain, which is the main user of NADH in the cell. When you do this, what would the highway look like? So you're really drawing the highway 3 times, okay? One, just like what are our rate limiting steps? 2, if we have ADP and NADH rising and dropping, what's that going to do? And then 3, what's going to happen if we inhibit some other pathway, okay? Again, with this larger view and picture, okay? Now, real quickly, actually, I'll just leave it at that. So pause it, okay, and do those things, and then come back and I'll give you some feedback, okay? Okay, so let's go ahead and look at this, and let's hope that you have, I'll try and keep both of these on the screen, but I'm going to have a little hard time doing that. But hopefully you're able to, quote-unquote, linearize this pathway, okay? And with that pathway, what we would have here is we would start at oxoacetate, okay? And it would be converted to citrate, and it would be done, that step would be done irreversibly. Okay, that's an irreversible step. Acetyl-CoA is required to do this. Okay, so it will feed into this, and that will make citrate, okay? And then citrate will be converted to isocitrate, and that will be done in a reversible manner. Okay, the enzyme is reversible. And then the next step will be the conversion of that to alpha-ketoglutarate. Okay, and that's going to make some NADH. And then there's another irreversible step. Alpha-ketoglutarate is going to be converted to succinyl-CoA. Now I'm not putting everything here. Okay, for instance, carbon dioxide is produced in both of these steps, okay, and some other things, but we'll just put NADH. And then what I'm going to do here is the rest of the pathway, okay, is going to be essentially reversible steps. Okay, so I'll just draw one big reversible step arrow here, and that's going to represent essentially going from succinyl-CoA all the way back to oxoacetate. Okay, and we'll just put OAA here. Okay, and understand that as we go in this direction, a small amount of NADH is going to be made, and a small amount of ATP or GTP is going to be made. Okay, and so that's what our pathway is going to look like. Now, given that, then, if we were to draw this as a highway or an interstate, then it would look very similar to our first activity, and that would be this. It would have this first step here that would be kind of, right, bottlenecked, and then there would be that expansion, and then it go back to bottleneck, okay, for those steps, and then finally it would be an expansion here. Okay, there we go, and here we go. Okay, right, and so this step here would represent this step here would represent this, these two steps here right there would represent that, and then all of these steps here would be represented there. Okay, and that's what our pathway would look like. Rate limiting steps are the irreversible ones, just as we discussed the last time. That in the case of the citric acid cycle, that includes the first and then the third and the fourth steps. Okay, very similar to our hypothetical pathway. In fact, this hypothetical pathway is essentially glycolysis, not glycolysis, but the citric acid cycle. Okay, all right, now the next question is this, and that is that, well, what happens if NADH levels are dropping and ADP and levels are rising? Okay, and this is where we need to start to think about well, what is the citric acid cycle doing, and how is the regulation of a pathway important for maintenance of what we call steady states in the cell? So there are steady state concentrations of NADH. There are steady state concentrations of ADP and ATP, okay? And these concentrations, as they start to get out of whack in the cell, if you will, then the cell needs to adjust in some way so as to either bring them back up into normal concentrations or take them down into normal concentrations, okay? And to maintain that steady state, okay? So if we come up here and we look at this, right? Each of these here, let's look at this, right here, here, and here. Each of those purple boxes represents the enzyme that catalyzes the first, the third, and the fourth steps of citric acid cycle. So citrate synthase is the first step, isocitrate dehydrogenase is the third step, and alpha ketoglutarate dehydrogenase is the fourth step. Right? These are the irreversible steps. These are the inefficient enzymes. These are the enzymes that are rate limiting. Okay, and what you'll notice here is that NADH. Okay, there's this little hexagon here in red, and that represents the fact that NADH is going to inhibit this step. And you also see here that there's ADP, which is a green circle, okay, and those are going to be then activating. Okay, green light means activate. Okay, and when you think about that and what's really happening here, and I could also look at this, right? ACP, right, is a red hexagon. Okay, and that means stop, right? These are going to be allosteric inhibitors of these enzymes. Okay, and when you think about the citric acid cycle, one of the main products of the citric acid cycle is NADH. Well, NADH will then go on to be used by the electron transport chain to make ATP. So if NADH levels are high and ATP levels are high, right, which would be represented here and here and here and here in the cell, then it makes sense that the flux through the citric acid cycle should be slowed down so that you're not overproducing NADH, you're not overproducing ATP, and to maintain that steady state level. Okay, ADP, okay, is going to activate. So if ADP levels are high, that would indicate that ATP levels are dropping and ATP is being used, which means that we need to upregulate this process, the catabolic process, so that ATP can be generated through the citric acid cycle. Okay, so when we look at this, then it says below is a figure illustrating the regulatory mechanism of the citric acid cycle. Using this graph, redraw the highway, assuming NADH levels are dropping and ADP levels are rising. Okay, so we're gaining an activator and we're dropping the levels of an inhibitor. Okay, NADH being an inhibitor. And so what's going to happen to this pathway? Okay, well, in that case, then we would expect these steps here, okay, here, right here and here, we'd expect those steps to expand, okay? Meaning that you would lose inhibition of them and you gain, quote unquote, more lanes of traffic, if you will. Okay, so this new condition would be something that looks like this. I'll go ahead and draw it using yellow, I guess, right? And what we would see here is this. It would look something like this now. Okay. Where our rate limiting steps are not so rate limiting anymore, right? Meaning that we would have some fairly wide lanes opening up here so that material can be taken through the citric acid cycle in order to produce the NADH, in order to make the ATP that the cell is needing. And the cell knows that it needs ATP because ADP levels are high. Okay, and NADH levels are low, and that means that, maybe there's just not a great enough source of NADH to make that ATP, okay? And so there then would be this increased flux through the citric acid cycle in order to maintain those steady states, okay? Now, in our next one, it says, okay, well, what would happen if you were to inhibit the electron transport chain, which is the main user of NADH? Well, if you take away one of the main users of NADH, that means that NADH levels are all of a sudden going to start to rise. And if NADH levels are all of a sudden going to start to rise, then you're going to get a decrease, right, in the flux or the citric acid cycle because NADH is an inhibitor of that pathway. So it's very possible in those scenarios that then, the pathway would look something like this, where our our rate limiting steps become even more rate limiting, okay, than what we had originally drawn them as. Because they're getting some sort of negative feedback, okay, and that's going to decrease flex to the citric acid cycle so that NADH levels are no longer rising, okay, or being made out of control, okay, which can be damaging to the cell. It can actually lead to significant oxidative stress if that happens, okay. So that's one of the scenarios that could occur, is that NADH levels would start to rise because the electron transport chain is inhibited. That's going to feed back. It's going to inhibit the citric acid cycle, and voila, this is what you'd have in response. Okay, all right. So all that to say, right, we can see here one of the major concepts of metabolism is the fact that metabolic pathways are designed in certain ways so as to allow for regulation, whether it be upregulation or downregulation at specific points, and they are very sensitive to environmental conditions. And that is in order to maintain steady states in the cell. So we adjust the flux so that then ATP levels or ADP levels or NADH levels or NAD plus levels can be maintained at fairly constant states. Okay? All right. I hope that was helpful to you once again. If you have any other questions, please let me know.